So good afternoon, ladies and gentlemen. Uh, it's a great pleasure for me to introduce our young and dynamic speaker for the second session, Dr. Subhashish Haldar, who is going to talk to us about a very interesting topic, the covalent magnetic tweezers, a new window to see biology. The talk, I suppose, will cover on the cutting edge research, covalent magnetic tweezers, a topic that lies in the interface of biology, physics, and chemistry. As a biophysics myself, I'm quite excited to hear from Dr. Subhashish. Okay, so our current speaker, Dr. Subhashish, who is among the few experts working on this field, is currently with Ashoka University and working as an assistant professor. He obtained his PhD from Indian Institute of Chemical Biology, Calcutta, in the area of single molecule biophysics. And after completion of his PhD, he went on to do his postdoctoral research with the prestigious Institute Max Planck Institute of Biochemistry, Germany. And later on, he shifted his base to Columbia University, United States. He has published his work in various top ranking journals such as Nature, Biochemistry, Anguchem, to name a few. And Dr. Subhashish has received many awards such as the prestigious Ramalingam Swamy Re-Entry Fellowship from Department of Biotechnology, India. I'm happy to let you know that Ashoka University is the only institute which houses the covalent magnetic tweezers, which was developed by Dr. Subhashish Haldar. Okay, so ladies and gentlemen, please join me in welcoming Dr. Subhashish Haldar to our faculty development program. The stage is yours, Dr. Subhashish, so you can start your lecture now.
Thank you so much, Dr. Chandran, for the nice introduction, and thank you so much for the organizer for inviting me in this session. So it's really exciting to talk about it. So it's kind of our development on the covalent magnetic tweezer, and we'll talk about it today, right? So I'm in Ashoka University. So today I will talk about covalent magnetic tweezer and how you can use it to understand biology or biological system. But the development of the magnetic tweezer involve physics, mechanical energy, and all these different kind of things. So let's start. So so very briefly, we have a mechanobiochemistry lab. In our lab, we have two kind of focus areas. One is that development of the single molecular techniques, and the second is that understanding of the complex biomechanics out of those. So I will give one example today, one example of developing single molecule techniques, which I will talk about real-time covalent magnetic tweezer, which will associated with the microfluidic system. And the second one is what the biology will understand from it. This will be basically with will describe with the mechanics of molecular chaperone. So now let first tell you that why you will be using this real-time covalent magnetic tweezer, right? You can ask me the question directly that what would be the advantage, why I should use that one. We have so many techniques. So the first one that is that with this technology, with a single experiment, you can get information about kinetics, that could be unfolding or refolding kinetics. I hope you all know kinetics from the background of chemistry and physics. So we can measure kinetics, we can measure thermodynamics, we can measure conformational change, how much the conformation is changing, we can check mechanical strength, and we can check polymer elasticity, all from a single experiment and from a single molecule. So a single experiment can provide you information about kinetics, thermodynamics, conformational change, mechanical strength, and polymer elasticity. So this includes most of the biochemistry and biophysics experiment. Secondly, with the advantage of that, you can see the changes of all the properties in a real time. That means that you want to see that what is the change of a kinetic property when you are adding some drugs or nanoparticle. So you can again see those changes in kinetics or thermodynamics in all these 5 molecular properties by just a single experiment. right? And with the same molecule. So I guess till now, monitoring all these properties in a same molecule is not possible with any of the single molecule technologies, which is the great advantage of this one, and which we are really, proud of. So the last point is that, but it's not the least, is that till now we are very happy that we can see a single molecule because this is a hot topic. But a molecule are basically based on several bonds, several atoms like that. So now we can actually resolute up to a single bond. So our resolution is not single molecule. We call our resolution is up to a single bond. So anyways, that, I also have a little bit time constraint for the discussion and other stuff. So I will not go into details for all the topics like detection of single bond. I may not, explain in the first phase, but if you are interested, and as I guess we have enough time, we can go and try to understand them, in the latter stages, right? So again, I'm summarizing it. Why you are actually using the covalent magnetic tweezer? Because a same single experiment will provide you unfolding kinetics, defolding kinetics, thermodynamics, polymer properties, mechanical strength, conformational change, all this information with the same molecule, with a single experiment. Not only that, you can check that how these properties are changing with with drug or biomolecules, that could be DNA and protein, that could be also derived from a single experiment, from a single molecule. And the last part, the not the least part, is that our resolution is single bond, not the single molecule. Great. So when we are talking about that, so let's see that, what are the technologies which are available in this field and what is the advantage of that. So if you know a little bit about force microscopy or force spectroscopy, you know that two technologies are really available. One is that AFM and one is that optical tweezer. So, and now I will tell you what is the advantage of our special covalent magnetic tweezer, which is really distinct from the magnetic tweezer which we have. So, so the first limitation of the AFM is that it's mainly monitoring the conformation on the high force range. So if this is the force range, so AFM is mostly monitoring above 50 piconewton. Whereas if you see optical tweezer is mostly focusing on below 50 piconewton. So our advantage is that with the magnetic tweezer, we can go from 0 to 100, like from the low force range, which is covered by optical tweezer, to the high force range, which can be covered by the AFM. So you can have a very high force range. Again, the second point is that with the AFM, you can see a particular molecule for a few seconds, like 5 seconds, 10 seconds, or 20 seconds. Same is happening with the optical tweezer. But with the magnetic tweezer, you can see a particular molecule for constantly 2 weeks. Yes, a single molecule you can constantly monitor 2 weeks. The advantage is that when you monitor a particular molecule for long time, maybe not for 2 weeks, but few hours, it gives you enough statistics to understand the fundamental mechanism of those processes. So now this is basically the instrument which we developed here in Ashoka. So this has several parts. So if you can see my mouse, you can see that. So it has one part is called voice coil here. Just the voice coil, there is a magnet, and just below the magnet where my mouse right now is basically the sample, the protein sample, which is lying down. And this is basically kind of the part of the instrument. And you see here is a CCD camera. So the point is that when I'm talking about the fluid chamber, it's kind of like that, which you can see here. So if you can see my face, you just need to see me, is that, here you have kind of a fluid chamber. So on the fluid chamber, you basically put the protein like that. This is my finger, which is moving. So this is my protein. And at the top of the protein, there is a small magnet, right? So what is happening is that you have a bigger magnet, which is sorry. Yeah. So we have a bigger magnet, which is N52. So the point is that you have a small magnet here, So when you are moving the bigger magnet towards the smaller magnet, it can actually feel the force, right? And this is the way you are actually applying force on the bead, which is top of the protein. And so if you are applying force on the top of the protein, it's actually the whole protein is feeling the force. And this is how we are controlling the force on the protein by changing the above magnet, so if you put it down, so it will feel the force, it will be differently react. If you took the magnet up, the force will be decreased, and this is how you are controlling the force. But some reason we are using not a single protein but several proteins together, and we are using copy pasting of 8 same proteins in the chamber. So now the whole story is the same. The protein is tagged to the glass surface here, and at the top of the place where my thumb is, here we have a small magnet, but here between the thumb and my last finger, little finger, we have actually 8 proteins of the same thing, right? Okay. So this is how the technique works. So you can see that this is the permanent magnet. Here is the permanent magnet, and we have a fluid chamber, which is basically here. So with the fluid chamber, we have all the chemistry of the single molecule, which I described to you before. And now you can see that you can really understand that when the magnet come closer, then the force increases. When the magnet is far apart, then the force is less. And thus we can actually plot in the right hand side. You can see that this is the magnet position and this is the force. So as we decrease the magnet position, that means as we bring the magnet down, the force increased, and as we decrease the force, the magnet should be going up. So how we calibrate the force, we can talk more, but this is kindly published in 2016 in JAX. So this is a real data. So this is excuse me. So this is a little bit complicated to explain the data. So it has several parts. So I will ask you to go very slowly with me. So here I will show you that how this covalent magnetic tweezer is mathematically controlled, or mathematically you can correlate that. So if you see at first you should see this force curve. So what is the force curve? This is basically very low force at 3 piconewton. Then you see I increase the force at 49 piconewton. Then I decrease the force, and the force is now kept at 6 piconewton. I hold the force for longer time, and at some point I pull it again at 49 piconewton. Right. So this is complicated, but here you can only check the force pulses. So what is happening when we pull the force from 3 piconewton to 49 piconewton? So I told you before that this is basically mathematically correlated, and we are using 8 of the same protein, right? Same protein copy pasted. So when you are pulling by all the 49 piconewton force, you can see that all the protein is basically unfolded one by one. And if all the protein is unfolded one by one, as we have 8 domain, we can count 8 steps. Let's count. So it is 1, 2, 3, 4, 5, 6, 7, 8, and you have before is the initial extension, right? So again, when you decrease the force, what do you expect? That you expect that proteins will now refold. So if we basically check this portion, then we can see that when we decrease the force, the protein is refolded one by one. As we have 8 domains here, you can again calculate all the 8 domains one by one refolding. So from the unfolding part, if you basically fit this curve with the exponentially, you can get unfolding rates. If you fit this curve exponentially, you can get refolding rates. So from the same curve, you have both the unfolding and the refolding. And now if I keep this position for long time, you can see the protein is hopping between the unfolded state and the refolded state. And for that one, you can actually calculate that at a fixed force, how the protein is hopping between folded and unfolded state, and that provides you the information about the equilibrium. So when we are talking about the equilibrium, that actually can provide me the information about delta G. Don't check these videos again. So, like, don't focus on here, let's focus here. So here the force is constant. It's basically 6 piconewton, and you can see that all the 6 piconewton, the protein is actually hopping, right? So here you can see that what is the folding probability. Folding probability means what is the most probable state where the protein could stay at this time. So here you see 8 domains is folded, then again 7 domain unfolded, again 8, again 7, then 6, 7, 6, 7, 6, 5, 6, 5. So protein is hopping like that. So you can here calculate that how long the protein is staying at each state. When you have all the protein is staying in the each state, you can literally calculate the delta G using the folding probability. So what is the folding probability? Folding probability is the state where protein wants to stay in the most of the time. Here roughly I can see that protein is staying in the 7th state. So that means it has 8 domain, around that 7 is folded. So that means 7 by 8 is its folding probability roughly, but the real calculation, you can actually calculate that what is the folding probability. So after that, getting the folding probability, you can calculate the delta G. So now in the previous one, we explain you that how you explain the folding probability, and you can get the information about delta G, kinetics, all this information. So maybe I could stop here once. If you have any question, please let me know. I would be happy to answer. If there is no question, then I will be going forward.
Yeah, Dr. Subhashish, I have one question.
Yes.
I'm Tyagesh here. In this thing you're talking about stacking the protein one over the other. So we can do this kind of experiments only with the proteins who can form kind of a heterodimers, something like that.
No, it's not. So I wanted to show you that how this is mathematically corrected. So here you can beautifully see that 8 unfolding, 8 refolding. So this is kind of a fingerprint. So that on the basis of that, you can see that here my protein is actually, it's a nice looking figure, but eventually I will show you with the figures with the one domain only, right? So it's actually a single domain. Yeah.
Okay, thank you.
Okay. Any other questions? Maybe not. So let's go forward. So, okay.
Sir, I am Akash from thermal engineering.
Yeah.
So my question is, sir, how optical tweezers helped in application?
So by the way, I'm talking about magnetic tweezer, right?
Yes, sir. Magnetic optical tweezer. Yes.
So this is magnetic tweezer. So right now I'm explaining you the technology, and in the second part, I will come to the application.
Okay, sir.
Okay.
Thank you, sir.
Okay. I guess any other question? No? Okay, let's go forward. So, I tell you previously that here with the single experiment, we can explain you that how all this information are changing with time. So now I will show you the same trace, but same trace means here the protein is in the low force. Then you unfolded that high force. Then you quenched it at 10 piconewton. So, it's 10 piconewton. The force is here. So the blue one is the 4 piconewton. You unfolded it at 50 piconewton, and then it gets quenched at 10 piconewton. So 10 piconewton, the protein is hopping between I guess 1, 0, 1 and 2 states. So you can see like 2, 1, 0, 1. So it's in between 0 to 1 state. And so it's 10 piconewton importantly. And then we unfolded it at high force at 50 piconewton, very similar way as I showed in the last slide. So then we exchange the solution. So here we are adding high salt concentration. This is not important whether we are adding salt or DNA or nanoparticles or protein, but we can actually change the solution in between the experiment. And again, we are tracking the same molecules. And now when we decrease the force, we see that a lot of proteins are refolding. And notably, again, you are monitoring the same molecule. And when you unfold it, again, you can see different unfolding steps. And when you pull it down, you can see that it's again 4 piconewton, and then you are applying force. So, it's the unfolding again. So let's check one thing. Here you can see that blue after red, that means 4 piconewton, it goes to 50 piconewton. you can see some unfolding. Here you can again see some unfolding that led to blue to red, that again the unfolding. So if we compare these two unfolding, then we can compare what is the unfolding rate at low salt and the high salt. Let's do that. So when we do that, we can see that this one, which is basically shown in the red, is actually taking longer time to unfold in comparison to that in the first thing. So that means is that here we can directly compare the unfolding kinetics of the same molecule, and we can see that the unfolding kinetics is much slower in presence of salt concentration. Again, here salt is not the important part for me. I just wanted to show you that you can see the difference of the all these things in the same molecule, right? So here you check, if you carefully, that you can see that there is a difference in the mean free passage time. Okay, so the first thing is that what is the mean free passage time? The mean free passage time we are defined as the minimum time to take from 0 folded domain to the 8 unfolded domain. So here there is the 0 unfolding, and here it is 8 unfolding. So this is the time the black one, the first one, which is basically in absence of salt, is taking, and then the red one, it will be taking a lot of longer time. So by eyes, without doing any analysis, any kind of tweaks, just comparing these two traces, you can see that how different they are with the mean free passage time. Then if you try to understand that, what is the change in the conformation, you can see that both the cases we are unfolding it from 4 to 50 piconewton, and if you are unfolding it at the same rate, you can actually see that although you are unfolding from the same post, there is a step size difference. So the step size difference clearly indicates that salt is affecting the conformational change. So again, if I compare these two traces, which is basically the folding part, I'm comparing this because both are happening at 10 piconewton. So if I comparing that, you can see that this one, which is basically much less folded in comparison of this one, which is much higher folded. So here there is 2 points. One is that kinetics is much faster with the red one. You can see that with your eyes that already 7 domains is folded up to this time, where with the red one it never reaches to the 7 folded. So unfolding kinetics is faster, as well as the you can see that the delta G, if you calculate the delta G, you can find out that the delta G is also decreased with the salt concentration. So again, my focus is not on the salt, but I want to see that all this information you can get it with the same molecule. Okay, again, I will stop again. So if you have any question, just happy to answer. Okay, so maybe there is no question, so let's move forward. So now, so what I promised that, with the same experiment, you can measure all these properties. You can see that same experiment, you can monitor unfolding kinetics, refolding kinetics, delta G, you can monitor the conformational change. And of course, with all this information, you can calculate the polymer elasticity. So if I move forward, so the next stage, what we are trying to understand that, okay, so with the same experiment, we can get all these properties, right? So now let's see that how these properties are actually, comparable if we do this experiment several times. So here is the B plot here where my mouse is right now, which is the B section. Here we check the unfolding kinetics at different salt concentration. And if you see the star, the the black star here and the pink star here is the value taken with the single experiment. So you see that all the data are really overlapping with each other. And if you remember that we found out that with the high concentration of salt, there is a small conformational change. So here you can see that this conformational change exists when we did the experiment with several forces, and we found out that they are actually for every forces with the high concentration of salt, the conformation is decreased. So that means the data what we found out with the same single experiment is quite quite matching with the results we found out with the many experiments together, right? Okay. So now the another advantage of the technique is that here we are not only doing the force clamp but also do the force clamp. So what is the force clamp and what is the force clamp? So force ramp is that where you are increasing the force at a constant velocity and decreasing the force at a constant velocity. So previously I showed you the force clamp. If you see my insert here, this is the force clamp, where you are increasing the force in a stepsize manner, but with the force clamp you are increasing the force in a constant velocity of up and down. So when you can do the constant velocity, you can actually get many information, like exactly you can check that what is the force where the protein gets unfolded, right? So you can get all the information, and this information actually helpful to get you that mechanical stability of the protein. So here, with the same protein, we basically pulse it several times, so we can run the pulse for several hours, even days. I take the 17, like with a 10-minute pulse. I showed you that with the each step, each place, you can calculate the 8 domains, like eight proteins, 1 2 3 4 5 6 7 8. That means that you can still see the 8 steps, whether you are doing with force clamp or force ramp. But depending on your expertise, what you want to do, you can actually do the experiments you wanted to do, right? So this is basically called force ramp, which provide us the information about mechanical strength. Again, I will stop for a minute. If you have any question, please go ahead. Yeah, so maybe not. So now let's come to the application part.
Sir, I have one question. This is Dr. Palash
Sure,
here.
please. Yeah,
Yeah.
please.
Sir, is there any limitation about the sample size or the kind of sample which is required for such kind of tweezers?
So so right now what you are seeing, all the data are taken with very small size protein. So so this protein which I'm showing you has 68 amino acid. So
Okay.
this is very relatively small. So 68 amino acid gives you a step size of 15 piconewton, right? So I can show you. Yeah, here is the resolution. So these are actually 10 piconewton, 10 nanometer. So 68 amino acid can provide you 15 nanometer, and we have the resolution up to 2 to 3 nanometer. So I guess with the difference of 10 amino acid, you can easily catch with this instrument, right? And if it is on the bigger side, like if the protein is bigger, it's actually easier to see it, because then you have the higher, longer step size.
Okay, sir. Okay, sir.
Yeah. Okay. Thank you. Okay, so now let's come to the second part, that okay, so we developed the instrument. So now the point is that what is the advantage of this instrument, like in what way we can actually use that. So I will explain all the advantage of the instrument with the with the molecular chaperoning system. So if you are not aware of the molecular chaperone, let me introduce that. So molecular chaperone are the most complex system in the cell. So you might heard that there are many proteins inside the cell which actually help us to doing several physiological activity. So as I know that few students are from the engineering background, so very briefly, so proteins are those molecules which actually help to do several physiological activity, like cytochrome C, they carry the protein from one end to other plate. You might heard about hemoglobin, so they are the protein which basically carry oxygen. So we have several proteins, means we have 20,000 different proteins in our body. So now the point is that this protein needs to fold properly to actively work. That means on a particular conformation, on a particular folding, your hemoglobin can carry oxygen. If hemoglobin cannot carry oxygen properly, then it would be an issue for you. Very similarly, you might heard about cancer. One of the protein which is basically mostly involved in cancer is p53. If the p53 is not working properly, you might have cancer, something like that, right? So now that we already know that the proteins are important, now the point is that why the chaperones are important. So chaperones are actually those part of the protein, those things which actually help the protein to fold properly. So previously we know that if the protein cannot fold properly, you could have disease. So chaperones are those machinery which actually help the protein to fold. I think you got it, right? Okay, so let's give you a brief discussion of how the chaperones work. We have several chaperone systems in our body. It starts with trigger factor, DNA-K, DNA-J, Seg-P, and then DSPA. We have several parts. So let's see something. So the protein is basically synthesized from the ribosome. When the protein is synthesized from the ribosome, it actually a polypeptide chain. That means it's an unfolded part. So DNA-K and DNA-J try to fold it. If they cannot fold it, they basically give the protein to another chaperone called Broviel. Broviel try to fold it. Very similarly, if DNA-K, DNA-J cannot fold it, sometimes it gives the protein to Seg-B. When Seg-B fold the protein, that is good. Otherwise, Seg-B is actually translate the protein to Seg-A, and Seg-A sometimes secrete the protein from cellular system to the outside system. So we all know that proteins required for cytoplasm. So this is the yellow portion is the cytoplasm. Protein is required for the periplasm or outside as well. So a lot of proteins needs to go out, and they needs to go out. They mostly go out with a channel, something like that. So when the protein is going out, the protein is actually here interacted with many molecules. So there is an important molecule which is called DSPA. So why DSPA is important? Because DSPA is introducing the disulfide bond. So one important point is also that you might know that in the cytoplasm, it is a highly reducing environment where there is no disulfide bond can form. But when the protein go to the periplasmic side, the DSPA is introducing the disulfide bond, right? So so this is kind of the known system with the chaperones. So now let's see, one thing is that what is the information which is known for chaperone. So we already known from the biology that chaperone is basically helping in protein folding by rescue, misfolding, or aggregation. So then we try to understand that it's already classified that chaperone could be two types. One is that holdage, one is foldage. Holdage means there are some chaperone which help the protein to unfold, right? And we already know that the holdage are trigger factor, DNA-K, DNA-J, they are the holdage, and the foldage is basically DNA-K, DNA-J. If you put the full system like DNA-K, DNA-J, group P, ATP, that's actually a foldage. So here you might find out a little bit confusing that here I put DNA-K, it is a holdage, and here when I put DNA-K, DNA-J, group P together, it works as a foldage. This is well known. I'm not describing that, but so if you put it alone, it works as a holdage. If you put the whole system, it works as a foldage. How they work as a holdage and foldage, I will explain. But the main point is that how the chaperones is classified. All the classification has been done in absence of force. So here in this project, we try to understand that how the chaperone will behave in presence of force. So the point is that why the force is relevant for chaperone. Why should see such biological behavior? So the point is that we know that protein may function under a force of 1 to 20 piconewton in several biological systems. That paper is coming out recently in Science, and one paper in Nature Communication, that protein can force under a force of 1 to 20 piconewton. So now the point is that we try to understand that how chaperones behave under force, and whether this classification of holdage and foldage could be changed in presence of force. So here I will give you a real example where you can see that how the chaperones are how the chaperones are interacting with the substrate protein under force. So okay, so this is the bacterial ribosome where a protein is coming out. When the protein is coming out, at first they interact with the trigger factor. So trigger factor, when they interact with the protein is just coming out of the ribosome and trying to fold. So when the protein fold at the edge of the ribosome in this place, due to the van der Waals repulsion, the whole protein is under a constant force, and Goldman et al. Monitor that the force is around 9 to 12 piconewton. So what we tried to do that with the magnetic tweezer, we can apply a constant force to the protein. So, we try to mimic that amount of force which the protein face when it interacts with the trigger factor, and we try to mimic that amount of force with the magnetic tweezer. When we did that, we found an interesting phenomena. So this is again a little bit complicated, so I wanted to focus only on this curve. So you see that I did the experiment in the same way, that at first I put the protein from 4 piconewton to 50 piconewton. We can see 8 domains unfolded. Then when we refolded back at 9 piconewton, we see only 1 domain refolded, right? So 9 piconewton is a high force. You see that in the last page, that this is kind of the force which is expected for the protein to experience when the protein is coming out of the ribosomal tunnel. So when 1 domain is folded, then when you put 50 piconewton, that single domain is unfolded again. And now when you add trigger factor, you can see that there is 6 domain refolded. So here with the same molecule, you can see that here only 1 domain folded, here 6 domain is folded, right? So you can easily calculate that, how much the folding is happening. So now you just imagine to think of a situation that I'm pulling your hand and you are also pulling your hand on your side. So that means is that I think you can feel that you are doing some mechanical work. So very similarly, you have to remember that we are applying force from the arrow this direction, and the protein is folding on the opposite direction. So when the protein is folding on the opposite direction of the force, it is doing some mechanical work. And this mechanical work you can easily calculate by multiplying force into the how much the protein is folding on the opposite direction. And if you calculate that, it becomes 59 zeptojoule. So there is an important point. Previously, you see that the trigger factor is known as unfolders, right? Which actually help in the unfolding. But here you can see that in the presence of force, it becomes a folders, right? So there is a marked difference. It's because trigger factor is previously classified in absence of force. So we thought that if trigger factor is behaving under force, let's see that how trigger factor is behaving in presence of force, and we found out that trigger factor works as a folders. So then we tried another chaperone, which is sec-p. So when we tried with the sec-p is the second chaperone which actually took the protein from trigger factor and carry the protein. We found out that sec-p is actually helping the unfolding process. How do you know that? If we check the mean free passage time of the refolding, we see that if the refolding time for the control is such less, sec-b is taking longer time to refold. All the rates are with the sec-b, where you can see that it takes longer time to refold. Very similarly, if you see the unfolding, you see that the unfolding is faster for sec-b. So sec-b is helping in the unfolding and slowing down the refolding. If we combine both the things, it will tell us that sec-b is actually helping in the unfolding process. So from the folding probability data, we can calculate the delta G. And if we basically keeping constant the energy barrier, we can find out that sec-b is actually helping the unfolding by shifting the energy barrier to the higher force range. So, so now the important take-home message is that sec-b is an unfolders in absence of force. Also, we found sec-b is an unfolders in presence of force. So then after sec-b, we test with DNAJ. I'm not explaining DNAJ because it is working very similarly as of sec-p, and we found out that it is basically unfolders in presence and absence of force. Very similarly, we found the same result with DNAK, that with the same mechanism, DNAK is working as unfolders, and it is working as unfolders in presence and absence of force. But there is a marked difference happen that, when we put the DNAJ, DNAK, QP, and ATP together. So the point is that DNA, so when you have the whole system, then it should work as a folders. But if you see that the curves, so it is not working as a folders, but it's working kind of a neutral chaperone. For example, the red one is basically with the DNAJ, DNAK with the group P, and the black one is the control. You see that all the curves are kind of very similar. I put it DNAJ, which is the unfolded, and you see that DNAJ is still working as an unfolded where the values are really low. But with this DNAJ ATK or control, you can see that the values are very similar. That means DNAJ ATP, the whole system is working as a neutral chaperone. It's not increased because it's almost correlating with the values of the control. But notably, in absence of force, DNAJKE is known as a strong folded, but in presence of force, it's behaving as a neutral chaperone. So then we try to understand another chaperone which behaves totally differently, which are basically shown with the DNAK or DNAJ. This shows that you can see that the control is here. So this is the refolding time for control, which is huge. Because when you put DSBA, you can see that the refolding is very faster, right? So that clearly shows is that now the refolding is very faster. And if we consider all these values, we can actually calculate the delta G, where we can see that the DSBA is actually helping the protein to fold by destabilizing the unfolded state, right? But previously it was known that DSBA is a weak folded. This is known when people did the experiment in absence of force. When we applied the physiological force range, we found out that DSBA is a strong folded. So now, like we try to understand why only the DSBA is working as a strong folded. So the reason is that actually how we did the experiment, DSBA has a thyroxine domain, right? So we are trying to understand that DSBA is a thyroxine domain. So that domain is actually working for the folded part. But the point is that we tried with the other cytoplasmic thyroxine protein, which are PDI, thyroxine, like every protein has the same thyroxine domain as DSBA. And when we did the experiment, we found out that DSBA is only working as a folded, but not PDI and thyroxine. So this also tells me that this is not something related with that thyroxine domain. So then why we try to understand that, we might thought that might be all the experiments coming from an artifact where you cannot get all the information of the same protein. So here with the experiment I showed you that we use a single protein, we apply a force of 9 piconewton, and what we see is that at 9 piconewton the DNAJ is actually least folded, whereas the DSBA is the highest folded. So if you do all the experiment with a single curve, you see that you can see that the difference with the DNAJ and DSBA. So and definitely you can calculate the mechanical energy. You can see that the folded protein, which is basically DSBA or trigger factor, which has the highest mechanical work, whereas the holded protein, they are the lowest mechanical work. So any questions from till now? Otherwise, I will go to the single domain protein, which I spoke about before. Any questions?
Yeah, Subhashish, I have
Yeah.
one question.
Sure.
In the previous experiment, what exactly was the control you used?
The control is used basically without any chaperones.
Any chaperones? Okay.
Yeah.
And, okay, fine. Thank you.
So we found out that few of the chaperones like DNAJ or DNAK, they basically unfold the protein faster from the control and refolding the protein slower than the control. So that definitely says that these are the holded protein. They basically take quicker time to unfold this. Right.
Okay.
So
Okay.
now we are explaining the whole point with a single protein, which we are talking before. Dr. Chandran was asking that what happened if the protein is a single domain. So previously we worked with a protein called protein L, which is mostly a beta domain protein. So now I'm talking about a talin, which is mostly alpha domain protein. So in the alpha domain protein, we did the experiment in the same way. Now if you check the B panel, you can see that how the domains are folding and unfolding. You can see that if you put, this is also a high force sensitive protein. If you check the 11.5 piconewton, the protein is mostly in the unfolded state. They try to jump to the folded state, but they cannot. If you decrease the force a little bit at 10 piconewton, you see that their population is same as the unfolded state and the refolded state. Now, if you decrease the force a little bit more, you can see that they're mostly staying in the folded state. This is very, very reasonable that if the force is less, it wants to stay in the folded state. If the force is high, it wants to stay in the unfolded state, and in between, it wants to hop between the folded state and the unfolded state. And this is the very similar the force lamp which we did to check the mechanical stability, which we basically described before. So we got a very same result which we found out previously, that when we have the DNAJ is an unfolded, we found out that unfolded is actually shift the protein towards the left hand side. DNAK is also an unfolded, so it shifts the protein towards the left hand side. DNAJK is a neutral protein. You can see that with the neutral protein, it's overlapping with the control. Here is the data. And for the folded one, now the protein is folding at a high force. Right, so very similar result with the taline protein as well, although I wanted to mention that both the proteins are structurally totally different. One is the beta sheet, one is the alpha helix. So then we can check the unfolding and refolding curves. From there, where we can get the Chevron plots, and from the Chevron plot we can see the same thing, that when it is unfolded, you basically get the unfolding force is the midpoint is kind of 9.8 or something. When you have the neutral, it is basically, I think there is a issue. This is a neutral, this is a unfolded. Sorry for that. This one is 10, and the unfolded is around 6. And when you have the folded, it basically show the midpoint at 15 piconewton. But the good part is that when we have this kind of data, you can fit the data with the Bell model, and when you fit the Bell model, you can see that the energy barrier, the unfolding is actually higher. Unfolding barrier is higher for the folded, whereas lower for the unfolded, whereas the folding barrier is higher for the unfolded and lower for the folded. So you can calculate all the energy barrier from a single plot. So now we try to understand, okay, let's think about something. What trigger factor and DSBA, they are behaving totally differently, a strong folded state when we are applying force. So what's the biological meaning of that? Why biology will come up with something. So the reason is that, when the protein is actually DSBA is presenting here, now you need to think about a translocation pole. So when the protein is coming out of the translocation pole, it is basically under force. The force is very similar of 9 to 10 piconewton. So the other point is that if the protein, if DSBA is not here, if it is not allow the protein to fold under force, what will happen is the backsliding and the protein will come down. But as DSBA is present here and it fold the protein with a high force, so it actually fold the protein, which actually easily shifts the protein from the cytoplasmic side to the periplasmic side. So this is how the protein is folded from the cytoplasm to the periplasm, and very similar mechanism happen with the trigger factor in the same place of the ribosomal tunnel. So on the basis of that, we basically classified the chaperones in 3 different parts. One is the mechanical folded chaperone, which actually associated with the tunnel, which is basically trigger factor and DSPA. This basically helps the protein, accelerates the protein folding against force. Then we have mechanically neutral chaperone. These are sometimes very fast folders in absence of force, but presence of force, they work as mechanically neutral. Whereas we have mechanically unfolded, DNAK, DNAJ, SECP, they basically transfer the protein. So they want to keep the protein in the unfolded state. So they are very same in presence and absence of the force. So yeah, this is kind of my publications from Ashoka. So all these comments which I basically said in my previous slides, you can find it all here. And this is my small group. So we are a few people working here in Ashoka, and thank you so much for the invitation. And if you have any question, please feel free to reach me on this email ID. And now this session is open for questions. Please let me know if you have any questions.
Yeah, Dr. Subhashish.
Yeah.
I was just wondering, actually, I was just wondering whether this can be used for this drug discovery one actually? Like, can we incubate the proteins with the drug molecules and
Exactly. So you are actually hit on the right spot. So you see that here you can check. So assume that, as I mentioned, like, you all know that P53 is a very important protein for the cancer. So what you can easily do is that, take P53 under force, check how the folding unfolding happens, and then you can actually pass several drugs one by one or in combination and check that which interact with the P53. So in this way, you can check the interaction in a very fast and small time.
okay? And what about any other protein? Like, not only sticking to P53, if you are interested in any other
Yeah,
protein
exactly. Any other protein
Mhm.
could be do in the same way. I'm just giving the example of P53
Okay.
because it's kind of well known. So, but any protein, like this is not limited to any protein. So you can check that particular
And
protein.
for docking the protein, you'll be going for this streptavidin, biotin. You have to label the protein by biotin, and then you'll be just uh,
Yeah,
incubating the protein over the streptavidin one, right? That's how
exactly.
you'll be
So
docking the
what we
protein.
need to do is that we have a
Mm.
particular construct. So we need to put the polyprotein inside the construct. So the polyprotein is one side is tagged with the avidin, where we can do the streptavidin binding, and other side of the polyprotein should be, halotag binding. Yeah.
Okay, thank you.
So we have a empty construct. So if we provide you the construct, if you put the protein inside the construct, so then that would be very easier to test all the proteins.
Okay, thank you. Because I normally I started working on the proteins purified from the natural source,
Yeah,
and some of the proteins may have a kind of a potential significance as the anti-cancer agents.
wow,
So I was
that
just
would be
wondering,
very
can I
interesting.
use as proteins, like study the mechanical properties of the protein?
Yeah, that would be great. Yeah, we can
And
definitely do that.
okay, thank you. And the second thing is like, can we understand the dynamics? Like nowadays, like we are using the molecular dynamic simulations of the protein to understand the protein dynamics. But I think with this current tweezers, maybe we can push a little bit further and do the actual physical study of the protein dynamics.
Exactly. So, the thing could be done in two ways, is that we can check the dynamics with the molecular dynamics simulation, and same thing, the dynamics with this instrument. And then that could be very good, like, comparison, or where you can, like directly compare the results from both the places. If they're matching, that would be great. Or if they're not matching, we can explain why those are not matching. That all are very interesting.
It's a very quite an interesting application. So, and the second thing is that this magnetic tweezers, the one you were talking about, because I just read the news that it is only available with the Ashoka University, and it's
Yeah,
not
it's
available
actually
anywhere in
available,
India.
yeah, in very few places in the world, like few places, 2, 3 places, I guess. Yeah,
So congratulations and for
thank you.
bringing such a cutting edge research to India actually. So is it open for the researchers if anybody is really interested in
Yeah. If
studying the
anybody,
samples?
so we have we have, so right now with the COVID and with some, uh, like we need some few more person. Otherwise, this is totally available. So this is available with my lab, plus, like, with the Ashoka University. So anybody who wants to do that could be easily done. Yeah, this is available.
Okay, thank you so much. Now we are just open for questions. If you have any quick questions for our speaker, this is the right time to have a kind of interactive session with him. So please unmute and just pop up your questions if you have any questions.
I have one small query actually.
Yeah.
Basically, I'm from the electrical domain, core electrical domain. So mine and yours research area is completely different. But anyways, such kind of characterizations which you have told in these tweezers, that we can do multiple characterizations from a single machine. So apart from the, I was more interested that suppose we have some solid materials or something like polymeric materials, something like that. So can we characterize those materials by the use of the machines which you have told?
Yeah, like I just need to know what are the materials. Like if they're
You can
polymer,
you
then
can
very
take it
easy.
as a polymer insulating material. Suppose you have any rubber material.
So the
It's
point,
kind of, yeah.
I got it. So the first thing is that, the biophysics, we are considering protein as a polymer. So, most of the, like there is a lot of group who are only study the polymer properties of protein. So, definitely any of the polymer could be studied, like, because a lot of people, we are consider protein as a polymer. So, of course, like, and I, as you are in the polymeric field, so as I spoke very briefly, but we are determining the length, counter length of the proteins. So, so those are the polymeric properties. So in place of protein, we can put also any polymer and then try to see how the properties are changing with force or with any interaction, whatever you want to do.
Okay, but only my concern was, Professor, is that with respect to the size, like protein size, what you have told, it's in the ranging of a nanometer, something like that. But I cannot make my polymeric material in the size range of nanometers.
Okay, so that part I don't know, but we have the limitation of 1 to 2 nanometer. I guess I'm quite sure if this is, like I'm saying, this is the lower limit, but not the higher limit.
I see,
So,
I see.
so this is the lower limit we can, like if the higher limit is like, if you have several hundreds nanometer, that would be also fine.
Okay, okay, okay. Thank you, Professor.
No problem.
Okay, so any further questions for our today's speaker? Any other questions are there? So I have one final question, Dr.
Sure,
Subhashish. Actually,
please.
yeah, this is regarding this. You very beautifully showed the protein folding and refolding the graph actually. So that I suppose that very much answers the protein folding funnel problem. Because we as biophysics, we have been always wondering like how the protein is like it's going for the self-assembly thing, because it's still a mystery for me at least, how the protein is folding and all. So in your talk, you have shown that by exhibiting a certain force, you can pull the protein and then it can refold back to its original form. So can you please comment on this actually? Because yeah, this is very intriguing for me. Like if you can pull the protein and we can just leave it, it can form its original bonds and it can regain its original shape also.
Yeah. So, like it's very easier to test it with this technology. So of course, so maybe not all the protein behave similarly, but you can take several kind of protein. Like here I took a alpha helix and a beta sheet protein. So here you can easily check that whether this is taking the same native state or not by checking the mechanical strength of the protein multiple times. So if we run the force lamp several times and found out that mechanical strength is not changing with respect to the first pool, that means is that if the first pool is the pool from the native one, so that means is that mechanical strength is not changing. That means it's coming on the native state. If the mechanical strength is changing with time or with the solution, then we have to think about that. Okay, this is not folding properly, so that could be molten globule, or that could be misfolding. So in that way, folding, misfolding, or this portion could be easily addressed by yeah, we could address that. Yeah, that could be very interesting.
Okay. Yep. So thank you once again,
Thank
Dr.
you so much.
Subhashish, for your wonderful session. It was a really an interesting and exciting session. Actually, we had a very beautiful insights about the cutting edge research which you are doing currently undertaking at Ashoka University. So from the participants and the coordinates, I take the pleasure in thanking you once again for your lecture. Thank you.
Thank you so much. Yeah, it's also great to talk here, and really looking forward to talk to you in future as well. If you have any question, just shoot me an email, and let's start our chat there. Thanks again, guys.
Definitely,
Very much,
Dr.
Dr.
Haldar. We'll be in touch with each other. Thank you.
Thank you. Bye bye.
Bye.